The next contribution of the morning is from Irena Ivanovska from UPENN. The talk will be Nuclear Rupture at Sites of High Curvature Compromises Retention of DNA Repair Factors. And if you are...
Okay. So I would like first to thank the organizers for giving me this opportunity to present part of my work. This is a work that I've done at Disher's Lab at University of Pennsylvania, where I also did my postdoc earlier, where I worked on mechanobiology of nucleus in stem cells differentiation. But here I'm going to talk about something rather different, although still in relation with the nucleus mechanobiology. I'm going to talk about nuclear envelope rupture as a possible mechanism for mislocalization of DNA repair factors and consequent excess of DNA damage. So there are many situations in vivo where cells have to move through constricted spaces in tissues, and when they do this, they have to squeeze their nucleus, and that causes a large deformation that might induce damage on the structural proteins that holds the nucleus together. And recently some labs actually did a lot of work on constricted migration in vitro, and including Disher's Lab, and they all show that with different assays that they see nuclear envelope rupture. There's always some bleb formation after the cell migrated through these constricted spaces. There's mislocalization of the DNA repair factors, and you can think in this very simplified way that if you have a sponge and that's your chromatin and you squeeze it, and then your repair factors are the water, it's just expelled out, and they segregate, which can lead to transient DNA damage. And also some work in our lab done with transwells show that if you have cells that will migrate many, many times through these pores, for example 17 in this case, there will be permanent genomic variation and also some phenotypic changes. So this work on constricted migration was done in Jan Lammerding's lab, but also at Mathieu Piel's lab in Curie in Paris, and also our lab. And there's other groups, for example, in Maastricht in Netherlands, there are groups who are working on DNA mislocalization and blebs formation, and probably we'll hear more later in this meeting about this. So here I stole an image of a breast cancer. This is actual breast cancer tissue from a patient from the Human Protein Atlas. So what you see here, these are the dark nuclei here. They show expression of the proliferation marker, which is used as a marker to identify tumor cells. And this is a good example that you can see that the nucleus are actually misshapen, and they have all these curvatures and probably kind of blebs formation in this tumor tissue. And the breast tumor tissue, we all know it's kind of a common knowledge that it's actually associated with pathological stiffness. And probably if you're a woman over 40 and if you go for your annual mammogram, you might hear from your technician that if you have very dense tissue in the breast, you better don't miss your biannual exams. So the reason for this is that cells exert forces via adhesions on their matrix, and usually they are tuned to their specific matrix stiffness, And if we have a pathological stiffness of like disease in tissue, then they have to exert higher forces, and those forces are transmitted to the nucleus, and the nucleus has to sustain it without breaking those stresses, and the proteins that are responsible for this are lamins. So here is a sketch that represents the nuclear lamin organization. They are two types of proteins, lamin A type and lamin B type, encoded by different genes. Lamin A is known to provide all the stiffness and strength to the nucleus, and it's not expressed in embryonic stem cells, but it's expressed in all differentiated cells. And lamin B is encoded by two different genes, and at least one of them is expressed in all somatic cells. So lamins, yesterday, I think, or a couple of days ago, we heard two talks that touch upon the lamins. One was by Alexander Zidovska, and the other was by Yana Feodorova, who talked a little bit about lamin involvement in chromatin organization. Lamins are involved in many other functions like DNA transcription, gene expression, but for the purpose of this talk, I'll concentrate mainly on the structure of the lamina. So there are intermediate filament proteins that form this kind of a meshwork from this, it looks like a juxtaposed polymer network. So you can see here high resolution imaging of lamin A and B. And this is the chromatin. And earlier we've done a work where we showed that lamin A is abundant in stiff stress tissue. Actually, it can vary over 30-fold. And we hypothesized that deficiency can actually weaken the nucleus, and it can cause structural damage. So how this meshwork looks like recently, a work that came from Medalia's lab in Zurich in Switzerland. They did cryo-electron tomography and gave us the molecular resolution of the lamina organization. So here, these are the nucleopores. You can see the density of the lamin filaments and how they are organized. So this is the nuclear envelope. So the important information that they gave us is that the thickness of this meshwork is about 14 nanometers, and the persistent length of the lamin filament is about 500 nanometers, which is something quite typical for intermediate filaments, but that makes it stiffer compared to the stiffness of the nuclear envelope membrane, which is much softer than this. So we hypothesized that in the case of high nuclear curvature, the lamin will resist bending, and this will lead to effective detachment of the lamin meshwork from the nuclear envelope, which will leave the nuclear envelope susceptible to rupture. So to test directly this hypothesis, first we perform AFM experiments on live cells that are expressing different proteins that are fluorescently tagged, and looked how they actually are mislocalized from the nucleus. So we used different probes, like probe with kind of a medium curvature in the scale of several microns, and also very sharp probe that is definitely less than 500 nanometers, which is the typical, the persistent length from what we learned from the lamin filament. And you can see here in these images that if we push with medium curvature, actually I had to mention how these experiments are particularly performed. So we indent the nucleus, and then we hold the probe for 100 seconds, and then the force is released. If you push very fast, like in an order of seconds, usually nothing can be seen even with a sharp probe. But in that way, with tip with low curvature, we don't see any mislocalization of the nuclear factor. And with a sharp tip, we see that within minutes, the DNA repair factors are leaking out of the nucleus into the cytoplasm. It's a very quick process. And we've done experiments with analyzed, but also with different repair factors like 53BP1, Q70 and Q80, who belong to a different pathway, so with the same or similar results in terms of time. And if we knock down lamin AC, which we speculated that has protective role, then even a higher curvature is, lower curvature tip is sufficient to break the nuclear envelope. So the question that we asked is this really nuclear rupture? Because it is known that if the nucleus was stretched, then the nuclear pores can be stretched, and there could be some possible passive transport from the cytoplasm or vice versa, from the nucleus to cytoplasm. So what we did dual transfection with a cGAS. cGAS is a cytoplasmic protein that binds DNA. It binds microbial DNA, but also, because it's not selective, it binds its own DNA. And you can see in these images here that when we push on the nucleus, then a few minutes after, we see the DNA repair factor leaking out, and there's a cGAS accumulation at the position of the nucleus. So then the question was, okay, is this a rupture actually only of the nuclear envelope, but something happens with the lamina? The next experiments that we did, we transfected cells with GFP lamin B receptor, which mimics the integrity of lamin B meshwork. And here you can probably see, so this is the M-cherry cGAS, which starts to flow into the nucleus, and here you can see there's a gap in the lamina. Okay, so this, of course, is a very kind of unusual geometry, and then we ask the question, can we prove this concept in an assay which is more closer to what we have the situation in vivo, not in restriction due to pores or external probes. And this is an experiment that was done on collagen, thin collagen film that self-assembly on atomically flat surface, and it forms this beautiful fibers where cells can adhere and start to elongate. So this is the system was earlier introduced by Daniel Müller in Dresden and co-workers somewhere in the early 2000s. So you can see that when the cells start to pull on the collagen fibers, they can squeeze it, but they also cannot really deform it in the direction of the fiber because it's much stiffer, so they're forced actually to spread. And what you see here in this image, these are nuclei, and if you have nuclei that is really stretched along the fibers with high curvature at the tips of the nucleus, usually we observe significantly more events of rupture and of mislocalization of the DNA repair factors to the cytoplasm. We analyze our data looking at the aspect ratio of this nuclei, but we also actually prove that the local curvature, the tips, really correlates with the aspect ratio of the nuclei in this case, and we also see that if we have a rupture, then at the tips there's accumulation of a cGAS, as you saw previously in the FM. Then we went to even simpler situation. We just used a stiff substrate and looked the cells that have high curvature. And what interestingly, we found that if there is a rupture, there was a depletion of lamin B protein, but there's also accumulation of lamin A. So it looks like that the lamin A is mobile enough and tries to go there to seal this hole. So I'll go a little bit more quickly through these slides. So this is the kinetic. Here the only message, important message is that the retention, the entry back to the nucleus of the repair factor takes many hours, and losing it, it's much faster process. It's less than 50 minutes. So now getting to the DNA damage, we have always some kind of a basal DNA damage in the cells. So we use gamma H2AX foci count as a marker of the DNA damage, and if we knock down lamin A, then we see increase of the DNA damage. If we overexpress it in these knockdown cells, we rescue the damage, and the foci number goes to the original basal level. So these are some data from experiments that we done with the idea of rescue. And what we did, we knocked down several repair factors, and then we overexpressed them to see if we can rescue. And only 53BP1 actually shows no significant difference, even if we knock down or we overexpress it. BRCA1 and QAT, definitely the overexpression can rescue the experiments. So this is something that I will skip for the sake of time. It's just about triple overexpression of this DNA repair factors. And I'm getting now to the effect of the matrix. So if the cells are on stiff matrix and then put on soft matrix, the curvature will be significantly reduced, so they will become more circular. And we did experiments to inhibit the myosin contractility, and in both cases we see that if we do this, we can reverse the DNA damage in cells. So finally, I just want to mention that we did some experiments in vivo where we engrafted tumors in mouse with cells that are lamin A knockdown and cells that are not. And after the excision and examination of the samples, we found that in case of cells that are not protected from the lamin AC, we see more cytoplasmic repair factor, and this is also reported for a different repair factor in breast cancer, for example. And with this, I would like to go to my conclusion. So what I show you today is that the nuclear envelope rupture is favored by high curvature, low lamin A, high contractility, and rigid extracellular matrix. Nuclear envelope rupture causes loss of multiple DNA repair factors, which results in increased DNA damage. And if lamin A is deficient, then DNA damage increases, which leads to mutations or senescence, apoptosis, and etc. And with that, I would like to acknowledge my co-workers, the professor I'm working with, Dennis Disher. Yuntaxi is a grad student who did a lot of work in general in this project, and these are all the other graduate students that are working currently on this project. There are two postdocs that contributed to, and we collaborate with Andrea Liu in the physics department. and Roger Grenberg in the medical school. And with this, I would like to thank you for your attention.
Questions?
Yeah, just some clarification. So the mechanism you're proposing is that the lamin essentially leaves the region of high curvature, so the sort of superstructure sort of melts in some sense around there, as opposed to what I would call a fracture process, whereby you break some region under high stress and then neighboring regions have even higher stress and you get a sort of avalanche of breaking. So that's not what you're claiming. You're claiming
Yes, so we have a lot of evidence actually that it is lamin AC is somehow accumulates at this position, and what we are working on is to see actually how this meshwork changes when one or the other lamins are knocked down. Is it really kind of overall change density of the change organization of these filaments, or is it like when the one is absent, then the other cannot really make this structure, this cross-linking between them? But the idea is that it's not an avalanche process, and we don't know really what is it.
I guess related just clarification. So a nuclear bleb is the region which is not filled with either lamin or filled with only lamin A. In analogy with the cytosol bleb, which would be not filled with actin. I'm just trying to understand what precise definition of a nuclear bleb is besides the microscope image of something that looks like a bleb.
So it is a formation that it has the nuclear envelope. It is filled with lamin A and lamin B, but at the position of the high curvature, the tip of this bleb, there's a mismatch of the signal between the A and B, and sometimes these blebs actually can break out of the nucleus and form this micronucleus, yeah. And they can actually also have DNA inside.
Oh, okay.
So, if there are no further questions, please join me in thanking the speaker.